TMS, transcranial magnetic stimulation. So we're moving from brain imaging to brain stimulation. And this is kind of a fundamental shift because up until now we've been talking about ways to actually look at brain activity. Here we're not looking at brain activity directly, we're moderating or modulating in some way brain activity, and then looking at the effects of that using some other kind of measure. So TMS research can't really exist on its own. It has to be combined at least with behavioral measures, and often with measures of either peripheral nerve activity, so recording motor evoked potentials, or behavior, or actually doing brain imaging simultaneously or immediately after, before and after TMS, to look at the effects on brain function or on behavior. There's an interesting history of brain stimulation that actually goes back to ancient Greeks. And I'm not really sure where somebody got the idea to put an electric fish next to somebody's head, or like an electric eel, but they did, and they did all kinds of crazy things back then. And it seemed to cure headaches, and so that was a thing. And then in the late 1700s, Luigi Galvani was the first to demonstrate what he called animal electricity, which was the stimulation of frog legs using electricity and showing that the muscles contracted when he stimulated them, suggesting that electricity was key to something about the way animals functioned, so calling it animal electricity. Around the same time, there were some attempts to cure blindness using electricity, and of course at this time electricity was not very well harnessed, but people were, it was very new at the time. That didn't go so well, but blind people actually reported seeing phosphenes, which are like the little stars you see if you bump your head or you stand up too quickly. So again, there's some evidence to humankind that electricity is critical in some way for brain function. Not very well defined. And then my favorite is Galvani's nephew, Giovanni, in 1803, would, quote, reanimate corpses. So he would actually show up at public executions with his stimulation device and wait till the person was nice and dead, and then zap them in and around the head and show that they would do things like twitch their arms or like make facial grimaces and so on, to the mixed cheering and horror of onlookers. That's what they did back then for fun. So again, some evidence that electricity is involved in some way. It's not super clear in that case, although he was stimulating the head. It's not clear that he was stimulating the brain directly as opposed to stimulating peripheral muscles that then caused limbs to dance and so on. But that was actually part of the inspiration for Mary Shelley's Frankenstein and the whole idea of electricity animating a monster kind of thing. Flash forward couple of hundred years. First stimulation of the motor cortex actually occurred in 1985, so relatively recently. And in 1989, the first impairment or disruption of visual processing. So sort of mid to late 80s is when TMS really became a thing as a modern sort of cognitive neuroscience research tool. Although, like most things, it took a while, and it was really sort of into the 90s and so on that it started to catch on, and more labs really got the hang of it and started using it, and we saw manufacturers starting to produce equipment that was dedicated for that purpose. Oh, I just want to jump back because this is another favorite picture. This is the one that, when's that from? I think sort of the mid 20th century. So that's actually all coils of copper wire. And so they were attempting to induce an electrical current in the brain using copper wire around and right-hand rule. And they thought if they wrapped enough copper around somebody's head, they might be able to do it. I think the problem there was that there was too much distance between the coils and the brain, and you need a very short distance. All right, so nowadays we have really two types of brain stimulation, both of which I'm going to cram into today's lecture. The first is TMS, which is the focus of the first part of the lecture, and the second is TES, or transcranial electrical stimulation. And in spite of the fact that they have very similar sounding names, just swapping out magnetic and electrical, they're actually completely different techniques in terms of not only how you do them, but the effects on the brain. And so TMS is induces quite a strong electrical current in the brain. I'll talk about how in a minute. And it's often described as inducing temporary lesions or virtual lesions, although it's not always disruptive. Sometimes it's facilitative, but it has very strong and immediate effects on the brain. Whereas TES involves passing very weak currents, so something generated by like a 9-volt battery. And it's generally, although it's sort of officially called stimulation, people really tend to call it neuromodulation rather than neurostimulation, because it tends to sort of bias neurons to be a little more likely or a little less likely to fire, but it doesn't cause the same level of disruption or stimulation that TMS does. So in spite of the similar sounding names, they're actually quite different in how they're done and in their effects. And I'll talk more later about the different flavors of TES. So TMS, the most sort of widely known, probably effective TMS is a temporary and immediate disruption of function. And actually I forgot to start with the video, but I'll show you a video at some point, maybe around the break, of what that actually looks like. But you stimulate over something like motor cortex, and you can get a twitch of the arm. If you stimulate over left inferior frontal gyrus, so Broca's area, and have people speak, like say the names of the week or something like that, you'll actually disrupt their speaking. It's not always total disruption. It's often more like sort of slurring of words or stuttering or something like that, but fairly obvious and immediate effects. You can also have alteration of function in wider networks. And so although TMS tends to be quite focal, we'll talk about how focal in a minute, it's a very strong effect. And of course, the brain is a big network. And so it's quite reasonable to expect that wherever you stimulate, the effects are going to carry through to all the different parts of the brain that are connected to that stimulated area. So you can have these wider ranging changes and changes that occur over longer periods of time, especially not with like a single pulse of TMS, but there are ways of doing it where you sort of stimulate repeatedly at some frequency, and those can often have effects that last for up to 30 minutes with one session, and even up to like weeks and months afterwards. In some cases, if you come in every day for like a 20-minute TMS session, over 10 days, that's a common treatment now for depression and some other kinds of conditions as well. So it can have effects over different timescales depending on the stimulation protocol. Can also be used for various types of cognitive enhancement and mood enhancement as well. So again, the effect of TMS on depression is sort of an elevation of mood, but you can get that even after a single session of TMS to right frontal areas, which is kind of interesting. Reduction of clinical symptoms, again, I just mentioned that. And it's state dependent, meaning that it's not just something that we put into the brain and it has the same effect on everybody. It actually is quite sensitive to things like whether somebody's physically active or has been physically active that day. They tend to have different thresholds or sensitivities to TMS than people who are less active. Just across individuals, there seems to be quite a range of the level of stimulation that's needed. So it is quite variable, and as well, things like different drug states and so on can affect the effects of TMS. So that's sort of big picture what it does. We'll talk more about specific examples as we go through. But the way it works is essentially relying on the right-hand rule. And so you have a coil, which looks like this, and inside that coil, there's copper wire running in a figure-8 kind of configuration. And so you pass an electrical current through, and it goes around, and there's multiple wind-ins. And so more and more wind-ins create a stronger magnetic field around the coil. And by the right-hand rule, that magnetic field is going to induce an electrical current in the brain when this coil is close to the brain. So essentially you touch the coil to the head, do this, you pass a large current through. The current is quite significant, and the field that's created is on the order of 1 to 2 Tesla. So that's sort of the similar strength to an MRI scanner. But it's very focal, right? So where an MRI scanner creates a very large magnetic field, but you have to have a shielded room and keep metal objects away from it. TMS, the field is very restricted, and so when you're doing TMS, you don't have to worry about metal in the room or things flying across the room and attacking you. It's quite safe in that respect. But it's still quite a significant current and significant magnetic field. And as a consequence of that, the hardware requires actually some charging. So you've got this big unit that generates the electrical current, and it actually takes a certain amount of time to sort of build up a big enough charge that then you press the trigger on the coil and deliver that stimulation. And then there's often sort of a refractory period before you can stimulate again. In some cases where you're doing repetitive TMS, the hardware is, you need extra hardware to sort of support that level of delivering current quite frequently. So it's quite significant that way. Oh, and the other thing I should point out here is the graph underneath is showing the electric field strength sort of spatially under that. And so the current that's induced is strongest directly under the center of that coil and sort of drops off fairly sharply with these two weaker lobes on either side, which are sort of the outside parts of the current. And keep in mind this is 2D, so this is like one slice through, but it's actually, you can imagine that kind of in a circle, right? So you've also got lobes kind of in front and behind that. There's different kinds of coils. All of them have copper wire inside. But there's different configurations that have different, basically, patterns of stimulation. So that figure 8 coil that I showed you is by far the most common one that's used because it induces the strongest effect directly under the center of the coil. So it's very sort of intuitive and easy to aim that and hit the right place. This one is simpler. I'll show you the different sort of patterns for these things in a minute. This one is designed to go a little deeper into the head. And this one, the big difference here, that big tube-looking thing is actually like a vacuum. So basically this is air-cooled, because one of the other things, because you're passing such a strong electrical current through this wire, the coils can actually heat up, and heat up to the point where it's uncomfortable to the touch. And so if you're doing repetitive TMS, especially over a longer period of time, having that air-cooled coil will keep it cool and allow you to be able to do it for longer than you would otherwise. Oh, and that up at the top there, that's the unit, so you can see the coil attached to it. The rest is basically the capacitors and stuff that are involved in drawing enough current and building that up to a point where you have the strong current that you can deliver. And so for the most part, coil orientation is something that people think about a bit less, but it's still important because the way the copper is wound, the orientation of the current that you induce in the head is aligned with the coil. That's the green arrow that you can see there. And so depending on how you hold the coil, you're actually passing the current in a different direction through the cortex. And so even targeting a particular area, you can find that by just rotating the coil, you can actually get stronger or weaker effects on the brain, just depending on the extent to which your coil and the current you're inducing is aligned with the neurons underneath. You can also see here that the current flow through that donut coil is a little bit different. And so that's shown in this next slide, where we're actually looking at computerized models of how strong the current is within the head, or in this case, sort of a spherical model of the head. So with the figure 8 coil here, you can see, so red is the maximum area of stimulation. So you can see again that it's maximal directly under, and there's actually sort of specifically weaker parts under the holes of each of those coils. Whereas with the circular donut coil, the strongest field is not directly under it, but really under the copper wire itself. So it's sort of a weird thing that you're stimulating, and you're stimulating the circle around the center of the coil, but not directly under it. So that's why the figure 8 is the one that's most widely used, because it stimulates one direct area, whereas there are relatively few cases where you'd want to stimulate everything around a particular brain area, but not that brain area itself. These two just sort of demonstrate that the size of the loops of the coil in the figure 8 have an effect. So the smaller coil gives you a more focal effect, but also the areas that are sort of weaker are much closer together. And there's also going to be a little bit less depth with that. The double cone, as opposed to the figure 8, it's basically just the figure 8 but sort of bent, and it's going to give you a bigger area of stimulation than the figure 8. And then there's these H coils. There's lots of other designs, but H coils are another one that's fairly common. And the motivation behind the H coil is to provide deeper stimulation. So you can see that most of these stimulations really directly sort of surface right where the coil is, or even in this one kind of around it. Whereas the H coil, you can see that directly under, there's relatively little stimulation, but the concentration is a bit deeper in the head. And so the idea of the H coil is, it was originally developed for studies, I believe, of schizophrenia, But the idea is that you can stimulate deeper parts of the cortex. So like in schizophrenia, they were targeting the orbital frontal cortex, so the deep sort of inferior part of the frontal lobe. And to do that, you need, obviously, to stimulate deeper. So that's the idea of these different designs of H-coils, is how can you make that sort of deeper and also relatively focal. To the extent that it's not focal, that's a consideration, obviously, because you can design something like one of these H-coils to target, say, orbital frontal cortex, but you can see it's actually stimulating kind of a big circle all the way around the sphere. And so you may not just be hitting the orbital frontal cortex, you may be hitting a lot of other areas. And to the extent that you're doing something like just trying to reduce symptoms of schizophrenia and it works, great. But to the extent that you want to make more interpretive or scientific conclusions about why it's working, you might have to be a little more circumspect, because you might actually be stimulating more than just the target area, and the effects could relate to that broader area of stimulation. And that can be hard to assess unless you have multiple coils, like this H1L is a more lateralized sort of stimulation pattern. So you could try that. One of the limitations is that all these coils cost money. And so unless you have lots and lots of money, you're sort of restricted by the number of coils you can afford to buy, and you're going to pick the ones that are going to be most amenable to most of the studies that you want to do. In terms of how you localize, the best way to do it is to use this kind of neuronavigation system. So in this kind of setup, you've got a computer with an anatomical image of somebody's brain. Ideally, it's the brain of the person you're trying to stimulate, because obviously that's going to be the most relevant. But barring that, because to get that, you have to put the person in the MRI. That costs money, takes time, etc., etc. You can use a generic, like a template brain, and you're not going to be quite as accurate, but you're at least going to be kind of in the right ballpark. And so you've got that, and then it's connected to this system using an infrared camera that's shining infrared light and measuring it. And then you've got the TMS coil, which has, it's a little hard to see there, but there are these little shiny balls that basically reflect infrared light. And you've got 3 of them in kind of a triangle configuration, all in a plane. And so just having those 3 points that define a plane, the infrared camera can find those and figure out the orientation and position of the coil in space. And you would also have something else attached to the person's head that has similar sort of reflective balls. So you have sort of a representation of where their head is and where the coil is. And then the neuronavigation system is smart enough to know, okay, given these balls and the balls are on specific points in the head, we can basically co-register, so align those with the MRI image and then tell where the TMS coil is relative to that MRI image. The other thing is that in this case, you've got this fancy chair with a mechanical arm that's holding the TMS coil. And that's pretty important, because if you just hold the thing with your hand, you're going to move around a little bit or a lot. And even just tilting the coil a little bit or turning it is going to affect the area of stimulation. And so if you want to be really precise and consistent, you really need the person sort of sitting in the chair in a fixed position, and then the coil held in place in a fixed position relative to them, so that you can deliver consistent stimulation. But given this kind of setup, you can see on your computer, not just on the outside of the head, but actually in the brain, where the TMS is targeting and how intense it's going to be. So you can target particular anatomical areas that way. And you can also combine that with fMRI. So if you've done fMRI on that individual, or if you have some sense from like a group fMRI study that you've done separately of a particular area you want to target, you can import those hotspots of activation into the neuronavigation and use your coil to target those areas specifically. So you can get some nice kind of precision there. So that's sort of the anatomically guided approach. The other way is what you might call, a second semi-anatomically guided way is to use the 10-20 system, so the same thing we use for locating EEG electrodes. And so there you're sort of less precise, you're relying more on assumptions that everybody's brain is more or less the same size and shape relative, maybe different sizes, but the same shape inside the head. And you can say, okay, if we're targeting the left motor cortex, electrode C3 is over the motor cortex. And so we'll just, sort of measure using our 10-10 system, find where C3 should be, and target that, or even use like an EEG cap without the electrodes in and target it that way. So that's sort of a coarser but much more affordable way of targeting than the neuronavigation, because obviously that whole system is going to cost you money. The other way is to use more functionally guided things. So we'll talk about motor mapping in a minute. So you can stimulate over motor cortex and find the part where, when stimulated, causes somebody's finger to twitch. And then you're stimulating the hand representation for that person. You can do it over the sort of occipital temporal areas and find somewhere that elicits phosphenes, so flashes of light, and that's usually around V5, so the motion sensitive cortex, and especially if those phosphenes are moving, that's a good indication that you're hitting V5. So if you want to study V5, that's one way to do it. You can't target every way functionally that way, though. Speech is another one, right? So I mentioned if you stimulate Broca's area, you'll cause speech disruption. So you can find areas, if you're interested in stimulating Broca's area, just by having people talk and do that. So those are the most coarsely grained ways in a sense, but they're also the most principled if your goal is to target your stimulation to an area that's defined functionally. So if I want to target somebody's speech area, it's actually a lot more reliable to say, I zapped around and I found the spot that caused the most disruption of speech, and that's what we targeted, as opposed to, based on a study of 20 other people who weren't this person, here's a blob of activation, and we just targeted that blob of activation, because in this individual, they may be, slightly less average, and so you might actually not be hitting the right place. Of course, you can use a hybrid kind of way as well. So that's how you sort of target different brain areas. And the stimulation is quite focal. I didn't mention numbers before, but it's usually on the order of like a few cubic centimeters. So relatively small, relatively focal, not hugely diffuse, except for the fact that there's like these network effects that the stimulation probably propagates to other areas. And there's different pulse shapes that you can use. And so these are very brief. So you can see the scale down at the bottom is 500 microseconds. So that's like thousandth of a second. Hundreds of thousandths of a second, so very short. The simplest pulse is this monophasic one, which is what people used for a while. It's actually more complex to generate because the default is you discharge and you get sort of a positive lobe and then the biphasic, so this sort of inverse lobe. And people initially thought, we just want to do one pulse and be done with it. Then they realized that these biphasic pulses actually induce stronger effects. And they also, it says, allows recycling of energy, because just the way the circuits work in the TMS, you need to generate, this big pulse, but then it can sort of oscillate. And so you can actually sort of harness that and recycle it back into the coil and cause continued stimulation. So get sort of stronger effects that way. And this is something that's maybe less obvious when you're reading sort of cognitive neuroscience papers about the use of TMS. But these pulse shapes can have a fairly significant effect on the strength and even some subtleties of the effects. And a big thing now is going beyond these very simple pulse shapes and actually customizing the shape of the pulse around particular principles or goals that you have. And there's one brand of TMS system that actually is designed to allow you to customize the pulse shapes. But again, the pulse shapes are a relatively minor thing compared to the stimulation protocols, which is what we're going to talk about next. So broadly speaking, these fall into, they're defined by the number of pulses and then the timing between the pulses. So the simplest is single pulse, so you just go boop and you're done. And this has certain use cases. There's some discussed in the chapter, talk less about it today. Because basically if you're only doing a single pulse, and if we look back at our things, right, we're talking 500 microseconds, you need to be pretty precise. So you can't say, start saying the days of the week and just do one super short pulse and get much of a measurable behavioral effect. But what you can do is if that's connected to a stimulus presentation system, you can do things like present a visual stimulus at very precise times relative to that pulse. And there's some cool studies that have shown that, for example, if you time the TMS pulse to line up exactly with person's N1 or P1 ERP component, you can cause them to actually not see a visual stimulus. Or you can disrupt, if you've heard of visual masking, which is where you present one stimulus and then a mask stimulus right after, very briefly, or even the other way around, you can get these effects where people don't see the stimulus where they would see it without the mask. And you can disrupt the mask effect through TMS as well. So again, precisely targeting it. So that's where you use single pulse, is really in those cases where you're aligning it and you're looking at something that's very precise around the timing, usually coupled with EEG so that you know how to do that timing. Paired pulse, I'll talk about more in a minute, but as it sounds, that's a pair of pulses fairly close in time together, and looking at how one pulse, an earlier pulse, affects the response to the second pulse a little later. So you can sort of amplify or reduce the effects of the second pulse depending on the timing and strength of that first pulse. Repetitive TMS is, I think, probably the most common in cognitive neuroscience, because there you're stimulating repetitively over a certain amount of time. So that's what would normally be used for something like the speech mapping, right, where you have somebody saying the days of the week and you can kind of move the coil around and it's pulsing the entire time. So you have this sort of ongoing effect. So that's used a lot, and the duration of the rTMS can have different effects. So sometimes that'll happen for like 10 or 20 minutes, and that has effects that can last for another 30 or more minutes afterwards. And then theta burst stimulation, which I'll talk about more, is sort of a variant of rTMS that people stumbled upon that seems to have effects like rTMS, but even longer lasting than that. All right, so single pulse TMS. The other case where this is useful is in doing motor mapping, and that's really where it was first really applied. So in motor mapping, you define some sort of systematic grid on the brain, ideally with your neuronavigation system, but you could also just, literally like draw these on with a wax pencil or something, or have your EEG type cap and have a bunch of dots on there. And you stimulate each point, and then you have an EMG connected to a particular muscle on the person's hand. So in this case, you can see it's on this muscle around the index finger. And with EMG, you're basically measuring the electrical potential between those two electrodes. And when you hit the spot in the motor cortex that controls that muscle, you'll get this motor evoked potential, or MEP. And you can get that even at levels of stimulation that would be below what would be required to really induce a real contraction of that muscle. So it'll contract a little bit, but you won't necessarily see the finger move. If you stimulate more strongly, you would be able to actually cause the finger to wiggle, which is kind of a weird experience if you've ever had that happen. So the idea is you systematically stimulate just using single pulses and look for a resulting motor evoked potential and find the spot that induces the biggest motor evoked potential. And then you're targeting that muscle group. And then if you're doing studies of the motor system, you would generally sort of continue from there by doing maybe paired pulse stimulation or other manipulations, looking at the effects of those manipulations on that, on that motor evoked potential. The other thing where this is used is in determining somebody's motor threshold. And so this is defined, typically what people use is the resting motor threshold, or RMT. And so this is the strength of TMS pulse that will induce motor evoked potential on 50% of the trials. So that's why it's called the threshold. So it's not 100%, but it's sort of right at the point where you're sort of at threshold. And RMT is used in mapping, in most TMS studies as kind of determining the subject-specific level of stimulation for that study. Because everybody has a different resting motor threshold, and as I mentioned, they can vary quite strongly from individual to individual, and especially if somebody's exercised or not recently. And so you don't define your stimulation in terms of like the actual voltage that you're giving to somebody and say, we're going to give everybody X volts of TMS, but you're going to do it as some percentage of the resting motor threshold. And sometimes it's like 80%, 90%, 100% of their RMT. And that way you're getting something that sort of across individuals has the same practical effect on their nervous system, right? So the resting motor threshold is thus called because people are they're at rest. They're not doing anything while you're stimulating. A variant is the active motor threshold, where you actually have them contracting the muscle, and that raises the threshold. So it's a different way of defining it that might be useful in motor studies where you're actually having people do motor activity. Another thing that people look at is that normally with EMG, just like with EEG, you've got background noise. So you have these electrodes on the hand, and even when the person's muscle isn't contracting, you're getting some sort of background level of noise just from random neurons firing in there. After you do a TMS pulse, there's actually a silent period where the EMG noise kind of flattens out. And it's like you've driven those neurons so much that they're all kind of refractory for a little period afterwards. And people actually use measurements of that, and both ipsilateral and contralateral. So remember, motor systems crossed. So if I'm stimulating right motor cortex, I'm going to get the left limb moving, or at least the muscles there stimulated. And so the contralateral, there's about the first 50 milliseconds of that silent period seem to relate to spinal mechanisms, so going down. And then there's cortical inhibition. And then you actually get silent periods ipsilaterally as well, which seems to relate to corpus callosum, these transcallosal kinds of connections. And so the silent period is actually a dependent measure in some studies, where they look at different things like if you're more active or less active, or other effects, how that actually modulates the silent period that you get after. So that's another measure there. Oh, I forgot this was still in here. So this is a nice example of using single pulse TMS in cognitive neuroscience. And so this is the one that I sort of alluded to already. So you have a target stimulus, and it's a letter detection experiment. So people basically just have to say if they saw these letters or not. And so you have a target stimulus that's on for 17 milliseconds. Any idea, by the way, why it's 17 milliseconds? Yeah? Nope. Good guess, but it's actually much more pragmatic than that. Think back to our lecture on psychopy.
Is it because if it happens less than 17 seconds, it's not possible that was coming from the...
Nope. Hint, it's not, there's no physiological reason.
It takes that long to read?
Nope. It's because, yeah.
Does this only do with refresh rate?
Yes, very good. Right, so your monitor refresh rate is 60 hertz, and if you divide 1 second by 60, it's 17 milliseconds. So that's the shortest that they could present a stimulus. That's all. Bonus points. All right, so they presented it for as short a period as they possibly could. And then they did a TMS pulse 100 milliseconds later, and then you had to report the letters. Then the other thing they did is a visual masking experiment where, again, target letters, then right after there was a masking stimulus, again, for as short as possible, and that was at 100 milliseconds and during the TMS, or sorry, the TMS was after, and then report the letters. And so there's two different experiments going on. The letter detection experiment is the blue line here, and basically what you're seeing is the proportion of times that they were accurately reporting those letters as a function of when the TMS pulse occurred. So they did the TMS pulse at different times relative to the onset of the stimulus. And what they found was that if the TMS pulse occurred in the first 50 milliseconds after stimulus onset, it was fine, there was no effect. But then there was this window between 50 and really 125 milliseconds where they were very poor at reporting the letters. And so, and this was delivered, by the way, to visual cortex, the TMS pulse. And so it seems that it actually takes about 50 milliseconds for the visual information to reach the visual cortex, and that's why the earlier pulses had no effect. And it turns out that this corresponds to like the P1 and N1 ERP components as well. Then you get better. And then the visual mask was kind of the inverse sort of thing. And so basically, remember there's the target and then the mask. Normally the mask without any TMS pulse will cause poor performance, which is why you see low proportion correct for that first 50 milliseconds. But if you TMS them in that 50 to 125-ish millisecond range after the mask, you disrupt the visual cortex registering the mask, and so you actually sort of unmask the original stimulus and see it. So it's just a really nice, elegant way of demonstrating how single pulse TMS can be used to assess timing of something like visual processing in a very fine-grained kind of way. And in that case, although I'm talking about how it aligns with P1N1, they weren't actually doing EEG within that study. So you can get, sort of very precise, what we call mental chronometry, without having to use EEG in that case. All right, paired pulse TMS involves a pair of pulses. They can be to the same brain area or to different brain areas, depending. And it's typically a conditioning stimulus, which is, so the dashed line here is the resting motor threshold. So the conditioning stimulus is typically either at or below the RMT, and then it's followed sometime later by a target stimulus, which is the one that we're really interested in looking at the effects of on behavior, MEPs or whatever. And here you can see, so as a function, so there's 4 different paired pulse protocols that are commonly used. These are short interval, short intracortical inhibition, long interval intracortical inhibition, intracortical facilitation, and short interval intracortical facilitation. And so they all vary both in the relative height of the CS and the TS and the relative timing between them. And you can see the effects on the MEP here. The first column is just the sort of the baseline, which is if you only got the target stimulus, that's sort of the default size MEP. And you can see that in the inhibition protocols, as the name suggests, you get a reduced MEP. And in the facilitation protocols, you get an increased MEP. So you can basically either sort of stimulate the system and cause kind of a refractory period, which is where you get the inhibition, if you have another stimulus coming in then. Or you can stimulate and then cause facilitation, so you're sort of priming the system, and so it'll generate a bigger motor response to the same stimulus later. And as I say, you can target these either to motor cortex or to different areas, and so you can look at things like, if we stimulate one area, does it have any effect on the effects of stimulating another area later? If it does, that suggests that those 2 areas are wired together. If it doesn't, it suggests they're not wired together. So that table's from the book. It's a bit much for a lecture, but basically there's sort of all these parameters. But the other interesting thing is that people have done pharmacological studies as well, where they give people agonists or antagonists of different neurotransmitters, like GABA, GABA-A, GABA-B, NMDA, glutamate, so inhibitory and excitatory neurotransmitters. And they've shown that these different effects are actually modulated by different neurotransmitter systems, relating to sort of the time courses of those different systems. And so what that's led to is this pharmaco-TMS EEG, where you actually give people some sort of drug and then do paired pulse TMS, and also you can record EEG responses to those TMS pulses. We'll talk about that a little later. And you can basically look at how the EEG responses relate to different neurotransmitter systems. So it's a way of sort of getting at pharmacology in a very brain-specific way that you couldn't get just by doing sort of give somebody a drug and measure behavior. Or potentially even just give somebody a drug and look at brain activity, because you're actually modulating or driving brain activity in some specific way. So for example, there was a study where they did paired pulse TMS in people with schizophrenia, and specifically using the LICI, so the long intracortical inhibition protocol, which is known to depend on GABA-B. They showed that people with schizophrenia showed a specific deficit in that paired pulse protocol, but not in other paired pulse protocols. And this was dorsolateral prefrontal cortex, which is part of the frontal lobe that's known to be affected in schizophrenia. And so it drew a link between schizophrenia, DLPC function, and specifically GABA-B, as opposed to other neurotransmitter systems. And in that case, without actually giving them the drug, just understanding from previous studies that that protocol related to that particular neurotransmitter system. We're sort of short on time, and so I'm not going to go into a lot of detail here. It's in the book, but it's kind of a neat study because they stimulated in 2 different areas, V1 and V5, and they were looking at information flow between the 2 areas, and specifically the fact that although information goes from V1 to V5, that's sort of the normal direction of flow, V5 actually feeds back into V1. And people had suggested that that was involved in the conscious perception of visual motion and sort of top-down processing. And it was already known that 20 milliseconds was about the time of sort of activation time difference between those 2 areas. And so they separated the TMS pulses by 20 milliseconds and looked at whether they stimulated V5 then V1 or V1 then V5, and showed that people's conscious awareness or conscious sensitivity to visual motion was only affected by that V5 to V1. So that basically supported this hypothesis that V5 was doing top-down modulation because it was sort of in one direction but not in the other. So that is another way in which you can use TMS to study time courses of information and sort of directionality of information flow, which is neat because in most cases, as I've talked about in the different neuroimaging lectures, with neuroimaging, we don't really have good timing information. Like with EEG and MEG, we can look at sort of gross levels of activation. But this is a nice way of really sort of causally linking these two brain areas and showing that the information is flowing in one direction and not the other. Or at least that there's particular kinds of information that then relate to behavior. Okay, so rTMS is where you stimulate repeatedly over some period of time. And there's sort of broadly two categories. One is low-frequency rTMS, where you're stimulating at relatively low frequencies, so 0.1 to 1 hertz. Remember, 1 hertz is 1 cycle per second. So 1 hertz is like 1 pulse every second, so it's pretty slow. 0.1 hertz is 1 pulse every 10 seconds, so that's incredibly slow. Typically, low-frequency TMS disrupts cognition, and effects can last for up to 30 minutes, at least if you do EEG to sort of look at alterations in brain activity. You can also do behavioral measures. It seems to selectively work on GABAergic neurons, which makes sense because those are inhibitory. It's an inhibitory neurotransmitter, and you're getting inhibitory type effects. And if you do that and then stick people in an fMRI scanner, they show decreases in cerebral blood flow in the areas that were stimulated. So that's sort of consistent with the inhibition idea as well. The other side of the coin is high-frequency TMS, which is sort of 1 hertz or above, but generally 5 or more hertz. Generally stimulates cognition. In both of these cases, it's not 100% always low frequency causes inhibition, high frequency causes excitation. Studies have shown sort of paradoxical effects, but by and large, that's the way it works. Again, about 30 minutes seems to stimulate glutamate, the excitatory neurotransmitter, and increase cerebral blood flow. So there's a fair amount of consistency around the effects behaviorally and neurologically. And then there's the theta burst stimulation, which has two flavors, either continuous or intermittent. So in theta burst, you can see, first of all, this timeline here over 30 seconds. It looks like just a whole series of pulses. When you zoom in and break that down, it's actually triplets of pulses separated by 20 milliseconds. So it's like did-did-did, and then a 200 millisecond gap, and then did-did-did, and then another gap. Whereas that goes on for a full sort of 30, 45 second period. The intermittent TMS is the same sort of triplets of did-did-did, but you go for, let's see, I think I have it somewhere. Let's see. 1, 2, 3, 4, 5, 6, 7, 8, about 10 bursts like that, and then a gap, and then another 10 bursts. You can see it like that. And so CTBS tends to be disruptive and inhibitory. ITBS is facilitative. Where these really stand out from the sort of slow and high frequency rTMS is that you get comparable effects to rTMS, but in a much shorter period of time. So with rTMS, if you're doing like an inhibitory protocol, you might do 20 minutes where the person's just sitting there getting rTMS, not doing anything else, and then you look at the effects after that on learning or some aspect of cognition. With CTBS, you can get the same effects in only 45 seconds of stimulation. So you've gone from like 20 minutes that they have to sit there not really doing much, just having their brain modulated, to 45 seconds. So it's a huge gain there. So that has caused it to become very popular, and especially that the effects as measured by EEG, but also by behavior, tend to last for like 45 minutes to an hour and a half. And so you actually have a longer window afterwards in which you can run experiments and look at the effects of the stimulation. So it's both shorter to administer and has longer lasting effects. So it's kind of win-win from a cognitive neuroscience perspective, and seems to, again, relate to GABA there. So in terms of how these things work, we know just physics-wise that we're inducing a current in a particular direction, and so it's probably going to induce that current in neurons that are aligned parallel to the direction of the induced current flow, because the neurons that run perpendicular to that, the current you're inducing just doesn't work, so you won't get anything. So it's mostly going to be, if you think about the alignment of neurons in the brain, there's some that are orthogonal to the surface of the cortex, like pyramidal cells, and then there's some that do sort of the lateral connections that run parallel, and it's those parallel running ones that are going to be most stimulated directly by TMS. And so these are actually largely sort of mostly local sort of connections between different neurons, whereas the pyramidal neurons are the ones that we think drive mostly like EEG and fMRI types of activations and do most of the cognition. And so it seems like in TMS, you're stimulating these lateral connections, and then there's indirect influence on the pyramidal neurons. And so this can affect their timing. And people generally talk about D waves and I waves. So the D wave is the initial effect of that stimulation and the current that you induce. And then the I wave, I for indirect, is after that you've stimulated those neurons, and then there's downstream kinds of effects of both those neurons stimulating other ones, but then there's also like refractory periods afterwards, because again, what you're doing with TMS is so much more dramatic and in terms of inducing the neurons to fire than what would be sort of biologically normal, that the refractory effects as well are quite prolonged compared to normal kinds of function. And yeah, again, they seem to affect both glutamatergic and GABAergic neurotransmitter systems. All right. One of the big issues with TMS is how do you control, right? So we can stimulate the brain. People know they're being stimulated. In fact, TMS produces quite a loud clicking sound and kind of a tap against your head. So people generally know when they're being stimulated. In some cases, maybe that's okay, but from like a strict cognitive neuroscience perspective, or even basic psychology perspective, we know that people are conscious entities that know that they're in an experiment and something's being done to them, and they probably expect that there's going to be some effect on the behavior of the TMS, and they might even have read the consent form, heaven forbid, and have some idea of what the study's actually about and what it's looking at. And so there's a potential, more in some studies, maybe less in other studies, of what we call demand characteristics. So basically the person performing either consciously or subconsciously in a way that's consistent with what they think the expectations of the experimenter is. And it can be really hard to separate those from the true effects of TMS if they're aware of when they're being TMS'd. So people have come up with a number of ways of doing sham stimulation that try and control for as many of these things as possible. One is just tilting the coil, because again, the current that's induced is so focal that if you just take the coil and sort of put the edge of it, if you imagine the coil would normally be like this, if I just tip it and put the edge against the head, it's still going to make the noise, and the tapping is because that current actually causes the whole, like the copper wire, to move, and so the whole thing sort of pulses a bit. So you're still going to get that tapping sensation, it's still going to be loud, you get the click sound, but the current won't actually be anywhere near the brain, and so you won't cause disruption that way. Another one that they do is stimulate directly over the vertex over the head, so like where your electrode CZ would be, on the thought that directly under that is actually just the central fissure between, or longitudinal fissure, between the two hemispheres of the brain, so you're just stimulating CSF and not so much cortical tissue. That's a bit of a strong assumption because obviously there's brain very close to that that could be affected. And in fact, people have shown that there is some diffuse but weak effects of vertex stimulation. Another issue is that you can blind the participant. It turns out that you can blind naive participants. It's very hard to blind people who have been in TMS studies before or people who work in a TMS lab, because they can still tell the difference between when the coil's on their head properly and when it's tilted. So it's not the best control. But you also want to ideally be able to blind the experimenter so that they're not behaving differently in the conditions where they expect an experimental effect versus when they don't. And that can be harder because obviously if the experimenter is doing the tilting or whatever of the coil, they're very aware of the manipulation. So the ideal thing is to use sham coils, which look like real TMS coils and produce a click noise and a tapping, but they have extra sort of design features that prevent an electrical current from actually being induced in the head. And so in principle, especially if they're not clearly sort of labeled as real and sham, you can blind not only this participant but the experimenter to which condition it is, especially if you have an experimenter who's like a junior research assistant or something who's trained but doesn't fully understand the whole system and so on. So that's the ideal thing, but the downside, the biggest downside of the sham coils is that they're expensive, and so it can be prohibitive for certain labs to be able to afford that extra hardware. but that's kind of the ideal thing. Other things people have tried is just stimulating, either, so the vertex is one sham site, but you could just stimulate in other parts of the brain that you expect not to be related to your task. And you could still get diffuse effects, but if your effects of TMS delivered to a particular brain area are as specific as you hypothesize they are, then stimulating other parts of the head shouldn't cause the same effects, even if it causes other kinds of effects. So at least it's a control in that way. But that's an important thing, and something that's not always obviously present in different studies, and it's worth, even when you see a control, thinking about what the control was and the extent to which there might be deficiencies because it's not double-blinded, or people might be able to sort of tell when it's a sham stimulation or whatever. Okay, so multimodal TMS. So you can do TMS with virtually any sort of neuroimaging system, and they're actually now like MRI compatible TMS devices, which is kind of neat because there's still metal in there, but like copper is not necessarily affected by the magnetic field, and so as long as you design the hardware right and have a really long cable, so the main part of the TMS is outside the MRI room, you can actually do it that way. Prior to that, people would do things like TMS somebody, so like the rTMS protocol, where you get lasting effects after 20 minutes. So you'd rTMS them, then quickly zip them into the scanner and start scanning right away. In this case, what they were doing was alternating fMRI scanning with TMS, because although TMS can go in the scanner, you can't actually scan well when the TMS is going because it's disrupting the electromagnetic field. So you kind of have to alternate your scans with TMS pulses. And in this case, they TMS'd different areas, including frontal eye fields, interparietal sulcus, so these areas are involved in visual attention, and then the vertex as a control point. And they looked at people's sensitivity to visual motion, and under a condition where you had TMS, a no TMS condition, and so on. So just in the interest of time, I'm not going to go into a lot of detail, but just to get a sense of how this can be done in an MRI setting. You can also do it with EEG, and again, you have to worry about the fact that the TMS is going to induce currents in your EEG system. So you can't actually recover the EEG directly when people are being stimulated, but you can get measurements directly after, or very soon after. And there's ways of designing EEG hardware so that you can make sure that, because besides just inducing big effects in the EEG system, first of all, those effects could be so big they really swamp the EEG system. So if you think of like when you're fiddling with electrodes, if you touch one of those electrodes, you'll get this big deflection that takes a little while for the EEG to come back down to baseline where you can record it. So same sort of thing happens with TMS, but you can build TMS-compatible EEG systems that will basically sort of gate the signal. So once the electricity coming through the wires hits a certain point, it just sort of shuts off and stops recording, so it doesn't overwhelm the system. What you're seeing here, the other thing that you can do is ICA, and that's what they've done here. So the red lines are the ERPs, so this is right after TMS, sort of during and after. And the red lines are the effect of the TMS, and then the black lines are after ICA correction. So you can actually remove the effects of TMS in some cases if they're not too big. And so you can look at the effects of TMS on ERPs evoked by different stimuli. Another thing people do is TEPs, or TMS evoked potentials, where you're looking not at like visual evoked potentials before and after TMS, but the effects that TMS actually induces. So when you stimulate somebody with TMS, you get a whole series of ERP components right afterwards that aren't the sort of TMS pulse being conducted through the EEG system, but they're actually generated by the brain as an effect of the TMS. And there's characteristic peaks that you're seeing here that are labeled that are not, well, the N1 is sort of a common one, but in this case it's a TEP. So these are all evoked by the TMS, and you wouldn't get them, otherwise, because the person's just sitting there. And each one has been linked to sort of a different synaptic junction. I think this is single pulse to M1. And so it's sort of different synaptic junctions within the motor system. And so you can actually sort of get a fair amount of sensitivity and then do other kinds of manipulations that you think would affect one or another sort of aspect of the motor system, and see if those are modulated by your task. So do the task, do the TEPs, see if they're modulated. So there's both the ability to look at sort of stimulus-evoked ERPs and how TMS might disrupt the brain areas that generate those. And then there's also the ability to look directly at the ERPs that are induced by TMS itself, especially in the motor system. And then finally, safety and ethical issues. The big issue here is that TMS can cause seizures because it's such a strong electrical potential. It turns out that this is not, it's not a non-concern, but it's a relatively infrequent event. And in the early days of TMS, there were a couple of big studies, or actually meetings, where they got together and looked at all of the reported cases of epilepsy or of seizures, and found that for the most part they were avoidable. And so people just weren't being careful about their inclusion-exclusion criteria for a study. And so it was people who had either history of seizures or family history of seizures or something like that. And so they published new ethical guidelines for doing TMS, and since those have been published, there haven't been more reported cases of seizures. So that's good. There's neuropsychological effects. So as I mentioned, TMS has been used to treat depression, and that came about because people noticed that when they did rTMS over right frontal lobes, people showed improved mood afterwards. Conversely, when they did it over left frontal lobes, people actually showed depressed mood. And so while it can be used as a treatment for depression over the right, if you're doing TMS over the left, you can actually make people depressed, which is a negative sort of neuropsychological side effect. And in other cases, we're intentionally manipulating brain activity, so you can have effects on memory and things like that. It can cause pain. Mostly the pain is because if you stimulate too close to like facial muscles, those facial muscles will contract. And in fact, doing TMS to Broca's area is close enough to the facial muscles that you get really quite annoying facial muscle contractions. I had a grad student who did a TMS study of prefrontal cortex for her dissertation, and she actually ended up getting bite guards for all of her subjects because their teeth were just chattering so much. That was with the CTBS, so it was only 45 seconds, but it was like 20 millisecond cycles of teeth chattering. It was quite unpleasant. You can potentially get burns, but that's pretty rare, especially with safety guidelines. There's some concern. So the idea of histotoxicity is that the cells, if you overstimulate cells, they can start releasing chemicals that are neurotoxic. It's more, again, of a theoretical concern. It's not something that's been demonstrated within the scope of kind of normal use cases of TMS. It's more if you push it to the extreme in an animal model, you might be able to see that. Effects of magnetic fields, so you don't want to TMS somebody who has, say, a metal plate in their head. Or a deep brain stimulator or something like that. And it makes a very loud, like a 90 to 100 decibel sound, so people need to wear hearing protection when they're getting TMS. And also not just the subject, but the operator as well. That's just a little more details of what I already said. And yeah, in the interest of time, I won't go into this in a lot of detail, but like any study, people should be provided with informed consent and fully understand what the risks are, where this is slightly higher risk than other kinds of cognitive neuroscience studies, and think about the benefit-risk ratio. And so even though TMS can cause seizures in people who are prone to seizures, it's actually also been used in studies of epilepsy and even in treatment of epilepsy. But the thinking is that if you're treating a condition and people are already suffering, the risk of inducing a seizure is maybe worth the risk, given that they might actually get benefit in terms of reduction of seizures, right? Whereas in a healthy individual who might have a family history of seizures, you probably wouldn't want to include them in your study if you're just doing some basic research, kind of cognitive neuroscience, rather than a clinically oriented kind of study.